quite some mails from people saying that they will be a little bit late. So I expect some people still to, to join us during the meeting. But anyhow, so welcome everybody to this webinar. My name is Mathieu Vinken. I'm a professor of toxicology affiliated with the Free University Brussels of Belgium. But today I'm here as the chair of the In2Tox Speciality Section, which is the in vitro and in silico toxicology specialty section of EUROTOX. I hope you're all aware of EUROTOX. So it's the overarching toxicology association and society in Europe. So we are quite active. We do have 5 specialty sections, and In2Tox is only one of them. So if you were to be interested to join any of these specialty sections, like this one, for example, please have a look at the EUROTOX website. You will find us in the About Us section. It is explained there what we are actually doing and how you can join. Besides this series of webinars that we have until the summer break, we also will have our meeting during the EUROTOX. Well, it's a joint EUROTOX and EUROTOX meeting that will take place 18 to 21 September in Maastricht in the Netherlands. So we have a meeting there. And we will also be, of course, meeting again in the EUROTOX conference that will take place the year after in 23, 10 to 13 September. And by the way, if you were to be of interest, we are still looking for session proposals, not only related to in vitro and in silico toxicology, but to toxicology as such. You can submit session proposals if you want for the 23 meeting. Please have a look at the EUROTOX website for this. So yeah, as I mentioned, we try to be quite visible, quite active. We set up this series of 6 webinars based on a survey that we held among the In2Tox members to see what they are really interested in and what they would like to see a webinar all about. So we already had a webinar last month, which was about artificial intelligence and how this is being used in toxicology. And in this very webinar, we will be focusing on AOPs, adverse outcome pathways. I think you already all heard about AOPs. They have been out there already for about 10 years, but there are still a lot of misunderstandings about AOPs, particularly regarding their, let's say, applications in the field of toxicology and risk assessment. So we are really honored today to have among us the speaker, for this webinar. And that is Penny Nymark. She is an assistant professor at the Institute of Environmental Medicine at the renowned Karolinska Institute in Sweden. She's also coordinator at the Swedish National Platform for Nanosafety. Her research and involvement in more than 10 European-funded projects over the past 15 years has focused on mechanisms of particle-induced toxicity and development of novel animal-free and fair data-driven methods, including AOPs for safety assessment of nano and advanced materials. So before giving the floor or the screen, so to say, to Penny, just some housekeeping information, if you will. This will be recorded. This is being recorded, and this recording will be available at the EUROTOX website afterwards. Of course, we like this to be an interactive kind of session, but to keep it a bit controllable, I would suggest that if you have any questions during the actual webinar, you can actually share this with me in the chat, and I will have a look at this at the very end. But at the end, we will have like a Q&A session, question and answer. So there you can then either, include your question in the chat, or you can speak up. And again, please, during the actual webinar, please mute yourself in order to avoid background noise. And with this, I'm very happy to give the floor or the screen, so to say, to Penny.
Thank you, Matthew. And thanks for inviting me here. I'm very excited to tell you all about AOPs. I think they're a really good thing and will advance toxicology. So with that, and you already know who I am, so I'm just going to go forward. Let's see if. Okay. So adverse outcome pathways are really something that I believe and many others believe are key in the ongoing transformation of toxicology. So this is a picture already published 14 years ago by Francis Collins, following the publication of the National Academies of Science report on the transformation or the move towards 21st century toxicology testing. And so what you see here, and let me see if I can find the pointer here. So what you really see here is that we are moving from, from animal testing, especially, and partly epidemiology, to understanding toxicity pathways better. And that's the basis, the underlying basis of really moving into 21st century toxicology. But of course, using toxicity pathways requires predictiveness and preferably causality between these mechanisms and the actual adverse outcomes in human beings. And the aim is really to move away from animal testing and use high throughput systems that are much quicker and keep pace with all the new materials and chemicals coming out. And that you, and that you would be able to prioritize towards animal testing if necessary. Preferably, you would never have to go into that, and then be able to predict human toxicity. So, and adverse outcome pathways are really the way to go in order to provide that causality between the key toxicity pathways and the adverse outcome, and to really retain that predictiveness. So today, 14 years later, this is a vision of predictive toxicology or transformed toxicology, which was recently shown by Nicole Kleinstroyer at the who is Deputy Director of the National Toxicity Program Interagency Center for Evaluation of Alternative Toxicology Methods. And she showed it at this conference on the Society of Advancing AOPs, where these things are being discussed. And what you see here is really a kind of circularity of the data and the information that we are generating over these or have been generating and developing over these past years. And a big part is really data. And I'm going to focus quite a lot on data in this presentation. AOPs really build on existing data, and I'll tell you more about that in a bit. And the aim here, so I will give you, so AOPs, of course, play a large role. They're only here in the corner, but they do play a really big role in determining or in having these mechanistic models that really provide us with predictiveness and experimentation towards safety assessment. And so I'm going to give you, so this is the basis of the presentation. I'll give you a bit of insight into what AOPs really are, going through a bit of conceptions and misconceptions about AOPs, what they can do. And then I will focus quite a lot on data integration and data, the usefulness of data and how to handle data, both for development of AOPs and a little bit on application of AOPs. And then I will also talk about FAIR data. And I'm hoping most of you have heard about FAIR, the FAIR principles. Even if you might not know exactly what they are, I'm going to give you a little bit of an insight into that, because I believe that matters a lot, both for development and application of AOPs. So, so the AOP concept is basically a way to incorporate mechanistic information into risk assessment. And what an AOP really does is we have had, so there have been concepts of mechanistic understanding within toxicology before. We've known this for a long time, that mechanisms matter, And it's better to understand the mechanisms than to have the black box animal that doesn't really, only gives you a readout of adverse outcome. Or today, of course, you can look closer at mechanisms, but it's always complex due to the complexity of in vivo situations. Now, toxicity pathways have, of course, been very central, and there have been aims to understand all the toxicity pathways that matter, basically. Now, they form a part of the AOP concept and are really the predictive components of the AOP. Now, then we also have the mode of action concept, which in fact is very different to the, it's very similar, but it's different to the AOP concept in the aspect that it contains the exposure part in the actual concept. So you're looking at chemical by chemical and connecting each chemical to a specific mode of action. Now, the AOP framework is really focused on the biology of the toxicity. So without it, they are stressor agnostic, and I'll get to that in a little bit. But that's one of the main differences to these previous concepts. So, okay, is someone saying something, or
yeah,
there's
please,
someone?
Please, can I please ask everybody to mute yourself, because there's still some background noise. If there were to be a question, please just share in the chat. Sorry, Penny, please go ahead.
Thanks. Okay, so AOP development has actually is guided basically by 5 main principles. So there's loads of guidelines and information on how to develop AOPs, but the 5 main principles that really matter and are important to keep in mind include the fact that they are not chemical or stressor specific. So they never couple specifically to one stressor. They describe the biology. They might describe some details relating to the stressor, but they are always stressor agnostic. And I'll also dig into that in a bit. The AOPs are modular and composed of reusable components. That's also a really central aspect, and I will also give you some examples on that. An AOP is also a pragmatic unit of development and evaluation, which means that it doesn't retain all the information about that biological process. It doesn't necessarily have all the networks and information that you would have to understand the complete biology. It contains pragmatic components that describe the very key elements of the chain of events towards adverse outcome. And so, I will get into that pragmatic aspect also in a little bit. And then the 2 last ones cover the fact that for real-world applications, AOP networks will probably matter. And I won't have time to go a lot into networks in this presentation. I hope maybe there will be follow-up presentations at some point or webinars to cover that aspect. But AOPs are also living documents, and I will touch on that a little bit in terms of and it relates basically to the circularity of data and the iterative process of improving toxicity or toxicology and toxicity testing based on the circularity of data. So now, importantly also to mention, the AOP framework is governed by the OECD, which means that it has a very high level impact in terms of regulatory practice. And the OECD has also developed a lot of guidance materials. So please have a look at that if you really are into developing AOPs, because it's very good to understand. These guidelines are in development, so they are flexible to further development. They are also living documents in a sense, and do take on new developments within the field of the AOP framework and within toxicology in general. So it's not a static guidance, but it's good to have a view of how AOPs should be developed. So, so overall, concluding on what an AOP really is, an AOP consists of one molecular initiating event, followed by a sequence of key events that are all causally linked through key event relationships. So these arrows are actually also, even though they're thin and small, they also contain a lot of information in addition to the actual key events. And then finally, one or sometimes several adverse outcomes. It depends on whether you're targeting organ level or individual level adverse outcomes. And what you can then summarize an adverse outcome pathway to is that it's a conceptual framework that portrays existing knowledge, important to remember here, concerning biologically plausible key events that causally link a molecular initiating event to an adverse outcome. And these AOPs are then stored in a repository called the AOP Wiki, which currently contains around, I think actually more than 300, but even close to 400 AOPs by now. And 19 of these AOPs are so-called OECD endorsed, which means that they are applicable to regulatory strategies or approaches. And they have been basically peer reviewed and accepted for actual official use, if you want. So now what can AOPs do then? What's really, you get the causal link between mechanisms and adverse outcome. Now, AOPs are developed through a series of steps. It's not really, you can't really identify what stage an AOP is at in terms of really defining the stage in very rigid manners, but an AOP follows this continuum of development through being putative, where you might have biological plausibility and strong evidence for the sequence of the different key events, but you might not have very high level confidence in the details of those key events. And you might, so it's not OECD endorsed at this level. You then move to a more formal level, which might become OECD endorsed, also called the qualitative level, where you really have a lot of detail and you understand the molecular players involved in the key events, and you have a good understanding of the relationships and the causality. And then following that, you can move into a quantitative stage where you have a very good understanding of the thresholds necessary for each key event to actually take place. So a type of, a quantitative understanding for each key event relationship. And these different development stages are applicable for many different types of risk assessment. So already in the early stages, putative AOPs can guide approaches for chemical or stressor, like nanomaterial prioritization. So when you see that something needs more testing, for example, or you might have a basis for grouping, for example. And much of these approaches are something that within Nanosafety, we call it pre-regulatory risk assessment, also coupled to, for example, safe by design strategies. And I don't know, I don't have time to go a lot into that, but that means that you are looking at safety of chemicals or nanomaterials already during the innovation of the actual chemical or nanomaterial or product applying these materials. So, and the early stages of AOPs can be very informative already at these types of risk assessment. And then moving towards the more formal and quantitative stages of the AOPs, they become more regulatorily applicable in, for example, integrated approaches to testing and assessment, IATAs, or even for defined approaches, which are less expert requiring than in comparison to IATAs, for example. So, so overall, of course, with new concepts comes also misconceptions. And AOPs have been, as Matthew said, they've been around for quite a while, but they are still misunderstood to some extent. And it depends a little bit on what field you come from or what type of data you normally work with. They are sometimes misunderstood to different levels. And there was a paper a few years ago where these misconceptions and kind of conceptions as well were presented and listed, and I think they're really good to take up. And I want to especially focus on the three first ones, which will be the basis for the next steps, but I'll go through all of them. So basically, AOPs are not signaling pathways. They are not molecular pathways that we see in life science databases that have, you know, extensive networks of interactions between genes, proteins, and other types of molecules. They are pragmatic. That's something to keep in mind. Pragmatic. They're not intended, closely related to this, they're not intended to be molecularly detailed. So they do have extensive amounts of information coupled to them, but they are not supposed to cover all the molecular details related to a specific toxic process. And they are not systems biology models, even though they integrate very different levels of biological organization. They don't provide systems biology in terms of relating to these other aspects as well. They are compound agnostic, which is, again, very important to remember. And surprisingly, even though it's one of the 5 principles, it's often forgotten. They describe toxicodynamics and not toxicokinetics. So they actually have to be coupled to model systems that predict or model for example, the exposure in different ways, ADME or PBPK models. They are multi-scale data integration tools. They allow for integration, and I'll get to that in the very end. And they provide plausibility, causality, basically, for statistical association within, for example, big data approaches. We know that anything can be correlated, and we often find mechanistic correlations with adverse outcomes, but the causality is not always a given. And the AOPs really provide that causality. And finally, they can identify knowledge gaps and indicate testing flaws and so on. I'm not going to touch that much on this in this talk. But so coupled to these first 3 points, that they are not supposed to be molecularly detailed, basically. It couples to this also recent paper on the fact that AOPs, so originally, we started off with these AOPs, which are pragmatic or which are described through a pragmatic simplicity. So people had been putting together AOPs that describe processes that were well known and well understood and coupled to a lot of detail, but not necessarily everything out there. And so we moved from this very pragmatic simplicity of these linear boxes. We all know also that biology isn't linear, but AOPs do stick to a linear model. Now, over time, we've moved a little bit towards the necessary complexity, because we do know that biology is complex. We have studied that for years and years and decades, and we know that there is complexity behind it. And now we're moving back towards simplicity, which allows us to apply all that information into, for example, risk assessment. And so we're moving here towards incorporation of that necessary complexity to then eventually move back to something that you would refer to as informed simplicity. I don't know why it keeps moving forward, my slide, but anyway, I hope you can stick with me. So informed simplicity is when you, for example, have a TV and you're watching the TV, but you don't know how it works behind it, but you can still use it and you can apply it, you can make TV shows, you can do a lot of things, but you don't necessarily understand the mechanics behind it. And that's basically what the AOPs are aimed at, utilizing all that background information that we have gathered over years, and then making it simple enough for different types of application. So, so giving you a little bit of examples from the nanosafety field. So as I said, AOPs are stressor agnostic. So this slide might confuse you a little bit, because then I say that nanomaterials are used as model stressors to develop AOPs. Well, for development of AOPs, you do sometimes take a strategy where you choose a type of stressor in order to develop the AOP, and then see whether it fits also other stressors. And this is what we've done within the nanosafety field, where we've taken data and understanding of nanomaterial toxicity and applied it to development of AOPs. So what you see here is a row of 6 AOPs that were developed, focused on lung adverse outcome, where one of them, actually the upper one, so I'm going to zoom in a little bit, or it's not zooming, but it's covering the other ones. So this one AOP, which is AOP 173, is one of the most well-developed AOPs within the nanosafety community. Remember, it's not only applicable to nanomaterials, it also fits other types of stressors, but originally it was developed based on data from nanomaterials, and more specifically titanium dioxide and I think carbon nanotubes to some extent. So it was developed by Sabina Halappanavar at Health Canada, and has been advanced quite a lot and is under review by the OECD expert groups. So basically what the AOP, it starts off with interaction with lung resident cell membrane components, which really is very typical for an AOP that fits or is applicable to risk assessment of nanomaterials, because nanomaterials in general have very non-specific interacting or initiating events. So this sometimes chemicals can be very specifically targeting specific receptors or other types of molecules, but in this case, it's really very general interaction. However, it does contain a lot of information, and there are more details on some molecular detail, even though it's not everything out there. But it does connect to different types of like toll-like receptor interactions and other types of receptors that are activated through nanomaterial contact. So, and then it moves through a row of inflammatory key events. It has a few associative events, which, however, I think in the final AOP are not currently considered in terms of measurability, but rather these ones here in this straight line are the ones that are actually part of the AOP. And then moving towards loss of alveolar membrane integrity, activation of T helper type 2 cells, fibroblast proliferation, and extracellular matrix deposition, which in fact is lung fibrosis. And so it basically has, and each and every key event contains information like this, and it contains, so you can see what type of information it contains in this table of contents here. And especially important is how it's measured. So each key event, a guideline for key events is also that they have to be measurable somehow. And if they're not, then there needs to be development of ways to measure these key events. So that's an important aspect of key events. Now, in addition, as I said, each arrow also contains a lot of information. And the key event relationship is really where you begin to build on the quantitative aspect of an AOP. So in this case, you see that the loss of alveolar, or sorry, the increased recruitment of pro-inflammatory cells leads to an increased loss of alveolar membrane integrity. And that then leads further to the next key event. And this is where this is hypothetical. It's not, it doesn't always look this perfect, but this is where you would have information on the thresholds required to kick off the next key event. So now, in addition to all this type of information that you have, as relating back to the pragmatic aspect and the informed aspect of the simplicity of AOPs, what is also important to remember is that we do need that complexity coming from life science databases that have been built over the last few decades, where there is complex molecular networks and interactions, and gene ontologies which describe the multitude of molecules that really interrelate in different ways. And so we have been together with a group at Maastricht University in the Netherlands. We have been looking at how to enrich AOPs with really the molecular details. So I'm kind of contradicting what I said before by not including all this information in the AOP wiki and in the AOPs, but linking to it and allowing it to be available for those who perform big data analysis, and so that the data circle can be iterative and inform the AOPs in terms of that complexity. So what we've done is map genes and pathways to AOPs in the AOP wiki, and then also connected different key events or events in an AOP to pathways that allow for big data analysis. So, and this, coupled to this, we've developed also a pathway, or we call it a data fusion pipeline, where we pull in information from these types of life science databases and connect them to in order to generate these interactive and bioinformatically useful AOP-linked molecular pathways. So, having that informed information behind the simplicity of the AOPs. And that then allows, so this is what it looks like. It's not meant to be read here. This is a molecular pathway that pulls in information from AOP 173 and allows it to be connected or allows an analysis to be AOP informed. So that basically what it then allows you to do in a risk assessment approach is to identify key biological processes and molecular players involved, like really to pinpoint those key events or key molecular players, and then allows you to develop broad coverage, high content assays like transcriptomics, for example, or other targeted high throughput screening approaches. And that then allows you to have so-called mechanistically anchored in vitro assays that are AOP informed. So kind of allowing for that big data analysis while having that causal connection from, or causal information from the simplistic AOPs. And that allows you also to report on the results from big data analysis in an AOP informed way, which allows people to understand it a bit easier, those who are not experts within big data approaches. So there's a row of approaches mentioned here that can be basically, doing grouping, for example, with an AOP linked information included, which means that you would group substances based on their potential to activate certain AOPs. Now, and this has actually been argued for in recent and new ways of doing risk assessment at the US EPA. So they recently published this blueprint for computational toxicology, where you would actually start risk assessment with high throughput big data generation, for example, transcriptomics, and then move into a type of hypothesis generation where you would utilize AOPs in order to pinpoint which AOPs, for example, which adverse outcomes might be in risk or related to the risk, and then being informed by those AOPs in order to select in vitro asset or select integrated testing strategies, for example, using in vitro approaches. And so these molecular pathways that are AOP informed really provide that basis for performing these types of hypothesis generating big data driven risk assessment approaches. So now I did want to, I'm worried I'm taking a lot of time. What time is it? I'm going to skip this part here, actually, and go into a little bit about the use of AOPs for risk assessment. So the data integration approaches that AOPs support, basically. So because mechanistic information, of course, and high throughput assays have had a hard time really fitting into regulatory risk assessment at the current state, because they are not, there's not a clear causal link between the results coming from these types of high throughput approaches and the end result or the adverse outcome. The AOPs really provide that, and that's what I've been reiterating over and over, that they provide this causality between the mechanistic aspect and all that data integration from life science information, that complexity that is necessary is informing these screening assays and informing the causality between these and the end point. But of course, there's also a lot of epidemiological, clinical data, a lot of legacy in vivo data that should be considered. And, with new materials, it doesn't maybe make sense to use old types of in vivo tests. But when it comes to grouping and looking at different types of approaches for novel ways of doing risk assessment, then it does matter to have legacy data included. And of course, we continue to do epidemiological studies. So the whole AOP basically allows for this type of integration of data over these different levels of biological organization. Okay, so it's jumping forward again. But so we did within Nanosafety, as I said in the beginning, we've been focused a lot on so-called safe by design strategies. And so I'm going to explain this picture just a little bit, where this bottom part here is something called a stage gate innovation funnel, which means that when you innovate products or chemicals or nanomaterials or whatever thing is being developed, you go through a row of steps in order to finally, so you go from an idea to a final launch of a product. Now, normally, traditionally, safety has been only taken into consideration just before launch. And mind you, these steps might take up to a few years to develop a product. And so when you go to launch, you want to test regulatory risk assessment and end up not being able to launch your product because you have something toxic involved. So the idea is that safety assessment would happen already early. And indeed, AOPs really, so this is a row of data approaches or data generation approaches, testing strategies that we have aligned along this stage gate approach. And AOPs indeed support that integration of data. So on many different levels, many different types of data, and many different versions and, types of data is maybe the best description. However, there's still challenges on how to really scale and score this type of data so that it really fits within or are comparable to each other. But AOPs are really supporting this type of integration. So I also had this, but I am really talking too much as usual. So I'm going to skip that and go to the fair data aspect. So in order to really develop AOPs, you need data. And you need data to be findable. You need it to be accessible, preferably also interoperable, and hence overall reusable. And this is something that really matters, especially in that circle of data use and data reuse in order to develop AOPs, but also in order to apply AOPs in risk assessment, because an AOP in itself doesn't do anything. You need data on a specific chemical that you're risk assessing, or on a similar, if you're doing a read across type of approach, then you need something that's similar. So fair data really matters, and findable, when it comes to being findable, it needs to include descriptive metadata. It needs to include persistent identifiers. It needs to be accessible so that someone can actually access it and use it, either through, it doesn't have to be open, but preferably it is. But if it's not, it might have to be through authorization, for example, accessible. Interoperable means that it's integratable, like, for example, having the AOP wiki coupled to other types of databases. That's an interoperability, for example. And it needs to be reusable, and that couples largely to licensing. So within the nanosafety world, we have done this for large sets of data. We've made the data fair, and we have discussed that in this paper here. And without going into detail on this aspect, those of you interested in nanomaterials might want to have a look at this database. But going to the AOP framework, Also, the AOPs need to be fair. So you need to have, not only does the data for the development need to be fair, but also the understanding of the AOPs. And that is largely coupled to the AOP wiki, which in fact is findable. It provides persistent identifiers for each of the AOPs and for many of the components in the AOP wiki. It provides accessibility to either through authorized editing rights or just open viewing rights. It does have interoperability because you can download the information and work offline with the AOP wiki information, which makes it interoperable with other types of bioinformatic approaches. And it does have clear licensing for reuse, which means that it's fair. So this was just a little bit on the importance of fairness within the AOP wiki and in relation to data for development of AOPs and application of AOPs. And so, but finally, I wanted to end with saying that in relation to that first slide that I showed regarding the other types of concepts that have been mechanistic concepts within toxicology, why are the AOPs now supposed to be so much better? Well, first of all, they provide causality. They are systematic, structured, quality controlled, and weight of evidence based. Now, you might argue that the other concepts have been that as well, but in relation to the fact that they are also governed by the OECD and hence picked up by large regulatory agencies across the world, means that this part really sticks to what it promises, that it's really a quality controlled effort. And finally, of course, AOPs allow for new ways of assessment due to the causality between the mechanism and the adverse outcome. So with that, I want to thank all my collaborators and all my funders. There's a load of them, so I'm not going to mention everyone. But it's all of this is enormous work by a lot of people. So yeah. And finally, I promised to also show a next webinar slide. So there's another webinar on 17th of March. So please look that up. Thanks.
Thank you, Penny. This was a very, very nice overview, very impressive work. So thank you so much for this. I don't see any questions in the chat. So I don't know, does any one of the attendees has a question now? You're allowed to speak up, of course. And you can also share in the chat. Okay, there is a question. So I will read it out loud, Penny. Mm-hmm. How to explain the gap between approximately 400 AOPs and only a few that have been endorsed by the OECD? How can you explain this? And how can you close this gap?
Yeah, good question. So as I said, the putative AOPs don't in general get OECD endorsement. And I would say a lot of them are still putative. They're not quantitative. Only a few, I think only the skin sensitization, perhaps the liver fibrosis one is getting a bit more quantitative. But overall, only one AOP is quantitative. And then there's a few more that are qualitative and have been endorsed, but it takes a long time to get them endorsed. So the AOP 173 that I showed is on the way, but it has been on the way, I think now for 2 years or something, and it takes a lot of work. So to really find the evidence and prove all the different small steps in an AOP takes a lot of time. So, you know, we're only in the beginning of this development.
Yeah, thank you very much. And I can witness this. I also made an AOP myself a couple of years ago, and it's a lot of work, especially if you want to do this in compliance with the OECD guidance. But the good thing about this is that, of course, I'm an academic myself, and I think that many of the attendees are academics. you always want to see some credits for the work that, or you get some credits for the work that you are doing. And in academic terms, this means a publication. And of course, this is a bit different compared to the outcome, or what actually OECD has in mind. But now they set up this very nice system together with now I think 2 journals, but they like to extend it to other journals. It's a co-submission system. So authors of AOPs would actually submit their paper, their AOP, to a journal, but at the same time, it would be revised, reviewed by the OECD. And of course, they are playing around a little bit still with the procedure, but in the end, it would be a win-win situation, because if it were to be accepted, then, as an author, you have a publication and it is included in the AOP database from the OECD.
Also, remember that putative AOPs are also useful for both, not only, so I said I focused on the pre-regulatory risk assessment, where you use new approaches like grouping or prioritization or other ways of just utilizing it for a pre-assessment. But of course, also for academics, I've heard a lot of stories where people really find them super useful in order to position yourself. If you're looking at one key event and you have a hard time, in your PhD, someone's asking you like, so how does this matter in the big scenario? And then if you have an AOP, it makes much more sense. It's easier to position. Also to identify gaps in research needs. There's a lot of applicability to research as well from AOPs. So you don't always have to go all the way to being OECD endorsed. They can be used for a lot more.
Absolutely. So I do have one question from my side, Penny. So you mentioned about applications of AOPs in the field of regulatory toxicology, so really for risk assessment. Now, this is a bit of still a controversial, let's say, topic. I mean, some areas are really open, to AOPs, others are really not. So what do you think it will take in order to convince the risk assessors to, using AOPs? And related to that, so what would you advise in terms of, efforts of quantifying AOPs? So how can we advance the field, basically?
Because I'm from a bioinformatics field, I see this extremely, I totally agree. And it's very difficult to get toxicogenomics to advance within the regulatory instances. We do see, however, an interest, but there's still confusion about the noise that toxicogenomics in general comes with, or so to speak, noise, because we don't see it as noise, but others might feel confused because there's so much information and it's too much. But so I would say that what is needed is, of course, as always, case studies providing, examples. And I think we do have more and more of that. And especially within Nanosafety, we actually, because nanomaterials still don't really have regulation. They're not covered, or they are covered by REACH regulation, for example, but there is still a big question mark on whether that regulation suffices. And so we do see some innovative approaches there. And I guess, it's largely about a cultural change, and we see that as well, that you just have to start using. And I think the AOPs support that, because it gives you that, it gives you that TV. You don't have to understand all the background, but you get a key event and someone says this is predictive of an adverse outcome, and you have that sequence of an OECD endorsed AOP, then a risk assessor starts feeling more comfortable in including that type of data. So, it's very complex, but it does, and these things just take a long time. And I guess that's the biggest step, is that we're now aware of the fact that it's needed. And the next step is to actually really apply in different case studies.
And there is a major role for us as scientists, of course, is to make this paradigm shift happen, because that's the most, you know, the biggest challenge. I see we have a final question in the chat. So I will read out loud. You spoke a lot about the upsides of AOPs, and I also think that they will be an important step for toxicology. However, I would be interested to hear if you see critical downside of the approach. So what are the disadvantages of using AOPs?
Yeah, good question. I wouldn't say that there's disadvantages, but there are some problematic areas. And that is maybe, and now I'm not super into the exposure side. I know in nanomaterial and nanosafety, the exposure part is very difficult to understand, and the transformation of the stressor in relation sometimes within the biology, and that's where we don't really have an answer of how that should be depicted in the AOP framework. So as I said, the AOPs are very biology focused, and I actually like that a lot. I find that very attractive to some extent. It's easier to keep away from the stressor. But of course, that brings the problem of what about persistent nanomaterials that stick around and cause frustrated phagocytosis or Ross cycles or things like that. And that's being discussed. And, you know, we just have to find solutions for that, or remember that the AOPs don't solve everything. They don't solve the need to understand what the end target organ is, or the transformations and the dose levels and all of that. So it's not a complete solution. But yeah,
that's true. It should not be used as a standalone tool. It should be used in, you know, together with other tools, so to say. Okay, I think it's almost 1 o'clock. I think we can wrap up. I would like to thank you so much, Penny. Despite having the flu, that you were among us, so please take good care of yourself.
Thanks.
I also like to thank the attendees, and also Elaine, of course, because she's supporting all of this, and she's logistically supporting all of this. And with this, I would like to invite you also to the next webinar, for which you have seen the ad already, which will take place the 17th of March, and that will be focused on kidney-based in vitro modeling. So with this, I'd like to thank you again very much. Again, the recording will be available on the EUROTOX website, and I hope to see you in March. Bye, everybody.
Thank you, and bye-bye.